On poursuit avec Olga Perez, Université de Montréal, qui va nous parler de Genomic Assessment in Microcystis, Coherence of Named Species, Geographic Signals, and Single Colony Sequencing. Olga?
Thank you so much. Hello, everyone. Today I'm going to talk about genomics in microcystis. Microcystis is a bloom-forming cyanobacteria. We can find microcystis in fresh water lakes. Also, we can find in reservoirs and lakes. Microcystis is unicellular. During the cell division, they had the tendency to make aggregates, forming colonies. These colonies are covered for exopolysaccharides or mucilates. Based in the cell site and the kind of mucilates and the morphology of the colonies, we can classify microcystis in different morphotypes or morphospecies. So in this case, also one interesting thing about microcystis is that microcystis can have attached in the mucilates heterotrophic bacteria. This is where we see microcystis in the real life. But today I'm going to talk only about genomics in microcystis. One of the main reasons to do this study is that a species or morphospecies definition is based in colonial morphology. However, we know that colony morphology can change. For example, we had two different morphotypes, Microcystis wesenbergi and Microcystis aeruginosa in the figure. We can see some difference. However, genomic comparisons show that microcystis morphospecies belong to a single species. For example, they did genome DNA hybridization, and they found that they had more than 70% of DNA reassociation that show the microcystis as a single species. Additionally, microcystis populations are thought to be dispersed everywhere with a homogeneous pool of genes. The main goals of this genomic study is to understand the coherence of microcystis species, the species definition in microcystis. Note that there's a biographic structure and look for presence of genes that are locally adapted. To start, we start with some isolated for, sorry, something wrong happened with the math. We start for the genome sequence of 32 isolated, 21 from Canada and 11 from Brazil. These isolated were initially characterized based in the morphology in 6 different morphotypes. Then, from these genomes, we resequenced 29 again in order to compare the presence of mutations between the initial genomes and the previous one. And also we want to know the presence of the change in the associated communities of bacteria in microcystis in the time. One of the main characteristics of the microcystis genomes is that they had general size of 4.6 megabytes, and also a GC content of 42. One of the main findings when we study microcystis using genomes is that microcystis appear to be a single species. But if we look deeper in the phylogenetic tree and also doing population genomics analysis using the core genome, the conserved genes in microcystis, we can see that there is some kind of clustering. For this, we got basis in the population genomic structure analysis, 8 clusters in microcystis, 8 genomic clusters. 4 of these clusters correspond to a species or morphotypes, like Microcystis paniformis, Microcystis flosaquae, and Microcystis wesenbergi. In the case, another thing is that if you look deeper in the clades, we can see that there is some kind of geographical signals. For example, the isolates from Microcystis flosaquae, they come all of them from Quebec. And also, for the case of Microcystis paniformis, all of them come from Brazil. What happened with Microcystis aeruginosa? One of the most studied microcystis. In this case, microcystis look be paraphyletic. That means that they are present in 4 of the clusters that we find. Another thing that I helped to understand the species coherence in microcystis, in the cluster, genomic cluster that we identify, is that we can see that within clusters, this is a matrix of values of homologous recombination. We can see that the intensity of colors is proportional to the recombination mutation rates. So we expect that within a species, the recombination values become to be higher, but between species, these values are lower. As we can see that within a species, like Microcystis paniformis and viridis, there is most recombination. This supports the species coherence in microcystis than between species. Another interesting thing about this is that after we look for the genomes in microcystis using the core genes, we start to analyze gene by gene what happens in every gene of microcystis. We start with the genes that are in the operon that encode for the cyanotoxins microcystins. So when we did a phylogenetic tree using one of the genes that are in this operon, we saw that 2 species are clustering together, in this case, Microcystis paniformis and Microcystis weissenbergii. That's meaning that probably in the same geographical region, they are recombining. And also, there is local adaptation. If we see here is the core genome using all the genes in microcystis, we can see that there is Microcystis paniformis and weissenbergii are in 2 different clades, but they are together when we analyze the one single gene. The interesting thing is that we see this in 5 of the genes that are in the operon related with microcystin synthesis. Another thing that we did analyzing the genomes of microcystis was looking for, we made a database of all operons related with cyanotoxin synthesis. In this case, we had a phylogenetic tree, and every horizontal line represents a microcystic genome. For example, in this case, we had from Brazil in orange and from Canada in blue, and the blue square represents presence of the genes, and white absence of the genes. We can see the red square that is showing that the complete operons related, for example, with microcystin synthesis. And we can see also that some microcystis or species of microcystis had encode for the complete operon of microcystin and microviridin. And also we can see that some of them doesn't have. So the idea with this is to try to improve the database, including more operons related with cyanotoxin synthesis. Finally, we did a new approach to study genomics in microcystis. The traditional way to study is culturing microcystis. However, the cultures take time. We went the last year to the Champlain Lake, and we take some colonies directly from the lake, and we clean these colonies, and then we characterize using the microscope, and then we did DNA extractions. The interesting thing about this is that we can't recover a big amount of colonies in a short period of time compared to the cultures that take time to grow. In this case, we had colonies, in yellow, the colonies that we isolated during the summer, the last year, in 6 time points. And also in purple, we had the colonies that will be sequenced. we did a successful DNA extraction from these colonies. As a preliminary test, we sequenced 3 of them. Fortunately, that was good because we got complete genomics only using colonies of microcystis, even with small quantities of DNA. The next step is try to move to another layers using the same technique, recovering colonies from the layer and then genome sequencing. Thank you. I want to say thank you to Shapiro Lab, Eve, Nico, Jesse, and also Julie, and especially thank you to Alexandra and Natalie.
I have a bit of a geographical question for you. My understanding is you said Microcystis flossicae is only detected in Quebec.
I said the genomes that we had, actually, we only have 4 Microcystis flossicae genomes. All of them come from Quebec, actually. And they, as shortest evolutionary time, you can see that they are together. So, but if you look in the database, in the NCBI, for example, there is not genomes with associated to Microcystis flossicae. The ideal scenario could be include more Microcystis flossicae morphotypes and see that they are distributed in other clades.
Okay, yeah, because I was kind of curious if flossicae has ever been observed in Brazil anywhere.
Yeah, it's true.
Microcystis flossicae observed in Brazil. Okay. Oh, okay, okay.